Hello, students. Welcome to Hello Biology. This is Lecture 2 of the chapter Principles of Inheritance and Variation.
In the previous lecture, we discussed genetics, heredity, and variation, Gregor Mendel and his experiments, why Mendel selected pea plants, Mendel's experimental techniques.
Today, we are going to study monohybrid cross in detail. We will understand monohybrid cross, reciprocal cross, dominant and recessive traits, Mendel's propositions, representation of traits using capital and small letters. So let's begin our journey into the fascinating world of inheritance. First of all, let's talk about monohybrid cross. What exactly is a monohybrid cross? A cross focusing on inheritance of one specific characteristic. In such a cross, only one character is studied at a time. A cross between two parents that differ in only one heritable character is called monohybrid cross, and the resulting hybrid is called monohybrid. Parents in a monohybrid cross are homozygous for the trait being studied. Now let us understand this experiment step by step. Seedling. First, Mendel selected two pure pea plants with contrasting characters. One plant was tall, and the other plant was dwarf. These plants formed the parental generation, also called the pea generation. Then Mendel crossed these two plants. After crossing, seeds were produced and grown into new plants. The plants obtained in the first generation are called the F1 generation, or first filial generation. An interesting observation was made here. All the plants in the F1 generation were tall. No dwarf plant appeared in this generation. This showed that one character was able to express itself completely, while the other character remained hidden. So, in this cross, the tall character appeared in all offspring of the F1 generation. Then Mendel allowed the plants of F1 generation to reproduce by self-pollination, and their seeds were obtained. When he sowed these seeds grew into tall stem plants and dwarf stem plants of F2 generation, giving nearly a 3-to-1 ratio of tall stem and dwarf stem plants. Most significant feature of this experiment was that dwarfness disappeared in F1 generation, but reappeared in about 25% plants of the F2 generation. F3 generation. Mendel allowed the plants of F2 generation to reproduce by self-pollination. The seeds produced by all the F2 plants were grown. These plants formed F3 generation. Mendel observation. Mendel observed that all dwarf stem plants of F2 generation were pure breeding. Out of the tall stem plants of F2 generation, only about 1/3 were pure breeding for tallness, while the remaining 2/3 produced both tall stem and dwarf stem plants in 3-to-1 ratio. Let's summarize the whole monohybrid experiment. Mendel crossed a pure tall plant with a pure dwarf plant in the parental generation, P. All plants in the F1 generation were tall. The F1 plants were self-bred to produce the F2 generation. In the F2 generation, both tall and dwarf plants appeared. The ratio observed in F2 generation was 3 tall, 1 dwarf. The same ratio continued in the F3 generation. Mendel did this for all 7 characters separately, one monohybrid cross for each character. This method allowed him to clearly track inheritance patterns for that single trait, without interference from others. Now Mendel started thinking very deeply. He observed that whenever a tall plant was crossed with a dwarf plant, all the plants in the F1 generation became tall. But one important question came into Mendel's mind. Does the result depend on which plant is used as male and which is used as female? To clear this doubt, Mendel performed another experiment called reciprocal cross.
In the first cross, he used the tall plant as one parent and the dwarf plant as the other parent. Then he reversed the parents in the second cross. This means the plant which was used first as one parent was now used as the opposite parent. After performing both crosses, Mendel carefully observed the results. And surprisingly, the result was exactly the same in both cases. In both crosses, all the plants of the F1 generation were tall. From this experiment, Mendel concluded that inheritance of characters does not depend on whether the trait comes from the male parent or the female parent. Both parents contribute equally in inheritance. This experiment helped Mendel prove that traits are transmitted equally from both parents to offspring.
Now let us discuss the results of the monohybrid cross. After performing his experiments carefully, Mendel made some very important observations. First, he noticed that certain, quote, somethings were passed from parents to offspring without changing. These hereditary units controlled the appearance of characters in the next generation. Mendel called these hereditary units as, quote, factors. Today, we know these factors as genes. Mendel also observed that the F1 plants were not intermediate between the two contrasting traits. For example, when a tall plant was crossed with a dwarf plant, the offspring were not medium-sized plants. Instead, all the F1 plants appeared tall. This showed that one trait was able to express itself completely, while the other trait remained hidden. Another important observation was seen in reciprocal crosses. Mendel found that the F1 plants obtained from reciprocal crosses were exactly similar. This proved that inheritance of traits does not depend on which parent contributes the character. Both parents contribute equally in inheritance.
Now let us understand another important result of Mendel's monohybrid cross. In the F2 generation, both the parental traits appeared again. This means both tall and dwarf plants were observed in the offspring. Now Mendel started thinking, if the dwarf trait disappeared in the F1 generation, then how did it suddenly reappear in the F2 generation? From this observation, Mendel concluded that the trait which did not appear in the F1 generation was not lost. It remained hidden or unexpressed in the hybrid plants. Mendel further explained that every character is controlled by two factors. Out of these two factors, one factor is able to express itself strongly in the F1 generation. This factor is called the dominant factor. The other factor remains hidden in the presence of the dominant factor. This factor is called the recessive factor. So in Mendel's experiment, the tall trait behaved as dominant, while the dwarf trait behaved as recessive.
Now Mendel carefully analyzed the inheritance pattern in the hybrid plants. He observed that there was no mixing of the two factors in the hybrid. The traits did not blend together permanently. Both factors remained separate inside the offspring. Then Mendel explained another very important concept. At the time of gamete formation, the two factors separate from each other and pass into different gametes independently. This process is called segregation. When these gametes combine again during fertilization, the traits reappear in the next generation. That is why in the F2 generation, both dominant and recessive traits were observed again. Mendel also noticed a fixed pattern in the F2 generation. The traits appeared in the ratio of 3 dominant, 1 recessive. For example, in plant height, 3 plants were tall while 1 plant was dwarf. This ratio is called the monohybrid ratio. Now students, carefully observe this diagram because it explains one of the most important ideas given by Mendel. Mendel noticed that when two contrasting traits were crossed, both traits did not behave in the same way. One trait remained visible in both the F1 and F2 generations. This trait was strong enough to express itself even in the presence of another trait. Mendel called this the dominant trait. For example, in pea plants, the tall trait appeared continuously in the offspring, but another trait disappeared in the F1 generation and reappeared again in the F2 generation. This trait remained hidden in the F1 generation because it was unable to express itself in the presence of the dominant trait. Mendel called this the recessive trait. Now the most important thing to understand is, the recessive trait was not destroyed or lost. Red exclamation mark. It was present inside the hybrid plant, but remained hidden temporarily. And when conditions became suitable in the F2 generation, it appeared again. So Mendel concluded that traits are controlled by factors, and among the two factors, one can dominate over the other. This simple observation became the foundation of modern genetics. Now, students, let us understand one of the most important concepts given by Mendel, dominant and recessive traits. Look carefully at this example of seed color. Mendel crossed yellow-colored seeds with green-colored seeds. After crossing them, all the seeds in the F1 generation became yellow. No green seed appeared in the F1 generation. Now Mendel started thinking, why did only yellow color appear? Where did the green color go? Then in the F2 generation, the green seeds appeared again. This clearly showed that the green trait was not lost. It was simply hidden in the F1 generation. So Mendel concluded that the trait which appears in both F1 and F2 generations is called the dominant traits. And the trait which disappears in F1 but reappears in F2 generation is called the recessive trait. In this example, yellow seed color is dominant, while green seed color is recessive. Similarly, Mendel studied 7 pairs of contrasting characters in pea plants. In every pair, one trait behaved as dominant, and the other behaved as recessive. Let us see these 7 contrasting characters one by one. Students, now we are going to study the 7 contrasting characters that Mendel selected in pea plants. Mendel carefully observed that for every character, there were 2 opposite forms called traits. Out of these 2 traits, one behaved as dominant and the other behaved as recessive. Let us understand them one by one. First is seed shape. Round seed is the dominant trait, while wrinkled seed is the recessive trait. Second is seed color. Yellow seed color is dominant, whereas green seed color is recessive. Third is flower color. Violet or purple flower color is dominant, while white flower color is recessive. Fourth is pod shape. Inflated pod is dominant, whereas constricted pod is recessive. Fifth is pod color. Green pod color is dominant, while yellow pod color is recessive. Sixth is flower position. Axial flower position is dominant, while terminal flower position is recessive. And finally, plant height. Tall plant is dominant, whereas dwarf plant is recessive. Mendel noticed one very important thing in all these experiments. Dominant traits appear in the F1 generation, while recessive traits remain hidden in F1 and reappear again in the F2 generation. This observation helped Mendel explain how traits are inherited from parents to offspring. Now, students, let us understand how Mendel's assumptions are connected with the terms we use today in genetics. When Mendel performed his experiments, the science of genetics was not developed, so he used simple terms according to his understanding. Today, modern genetics uses more advanced scientific terms for the same concepts. Let us understand this one by one. First is the term character. A character means a heritable feature. For example, seed color in pea plant. Seed color itself is called a character. Now, a character can exist in different forms. These different forms are called traits. For example, seed color can be yellow or green. So, yellow color and green color are traits of the same character. Mendel observed that these traits are controlled by certain hereditary units. He called these hereditary units factors. Today, we call these factors as genes. And the different forms of a gene are called alleles. So remember carefully. Character right-pointing arrow. A general feature. Trait right-pointing arrow. Different forms of that feature. Factor right-pointing arrow hereditary unit proposed by Mendel gene. Right-pointing arrow modern term for factor allele. Right-pointing arrow alternative forms of a gene. For example, seed color is a character. Yellow and green are traits. The hereditary unit controlling this is a gene. And yellow and green forms of that gene are called alleles. This is how Mendel's assumptions became the foundation of modern genetics.
Students, now let us understand how Mendel represented traits in genetics. Mendel needed a simple way to represent hereditary factors during his experiments, so he started using letters to represent traits. Now, remember one very important rule carefully. Dominant traits are represented by capital letters, and recessive traits are represented by small letters. For example, let us take seed shape. Round seed is the dominant trait, so Mendel represented it by a capital letter R. On the other hand, wrinkled seed is the recessive trait, so it is represented by a small letter r. This means capital letter equals dominant trait, small letter equals recessive trait. Now, look at the example shown on the slide. R represents round seed, which is dominant. R represents wrinkled seed, which is recessive. Mendel used this method to make genetic crosses simple and easy to understand. So, students, always remember this golden rule in genetics. Capital letter for dominant trait, small letter for recessive trait.
That's all for today's lecture, students. Today we understood monohybrid, cross-reciprocal, cross-dominant, and recessive traits. Mendel's propositions, representation of traits using capital and small letters. I hope this lecture helped you understand the concepts of inheritance in a simple and clear way. Books keep learning, keep growing, and don't forget to thumbs up like speech balloon, comment sent mail, share bell. Subscribe to the channel for more amazing biology lectures. Thank you.